Here, yeah. Is it like a 3-second delay? All right, so we have a couple absences today, so we're going to live stream today's class. We're going to start by talking about the BLAST lab. For those of you that are out there in AP World, the BLAST lab I have that you're going to see is on my YouTube channel, and a copy of this lab map, I believe, is also in the, what do you call it, the part underneath? The description. There's a link to this, so if you need this for your class, if that would be helpful for you, you just go right on ahead. Okay, we good? Am I on? Is anybody checking?
Oh yeah, one person's on.
Somebody check. I'm on it. Okay. All right, so remember I'm going to be gone next class, yes? Okay, so on that day, I want it to be very useful for you. I'm good. Okay, and that is you're going to be doing a lab. It is a digital lab. The whole time it's on your computer, so have your charged computer. You're going to do one copy of the lab per team. One copy of the lab per team. Did you hear me? Mm-hmm. And remember, whoever opens up the document and shares it, they're the one who needs to what? Turn it in. Because that will give me the record of your work. So if we are a lab team and I'm like, oh, I have to leave 7th period, I'm out. And when I leave, I'm the one who was logged in and I don't submit it. And she's like, oh my gosh. And then she makes a copy of it and submits it. It will only have her work on it. Do you understand? And then the rest of the members wouldn't get their work. So just whoever takes on the, did you lose something, sweetheart?
I can't find my glasses.
Oh, remember how I had a pair of glasses here for a while? And they are picked up.
I came in yesterday that I found at the lab table, ironically.
Oh, okay. I don't know, my dear.
Black and big and round, so just in case anyone.
Okay.
Can you see Emily's face?
All right, we'll try that.
Out of all the
Oh, when you turn around.
Every time it's just it's just like huge.
Okay, Emily is beautiful, and so she's on this video. It's an awesome
Yeah.
thing. All right. Sorry, we're distracted. It's Friday, and it was a rally. Do you want me to show them you? Okay. Yeah, do it. Just turn it around. I don't know. Like 3 people. Wait, make sure you're in the picture.
You can't even see it.
Go down.
That's my forehead.
Here's your bio buddy that you're missing.
I love my forehead. Oh.
And we're back. So anyway, I want to show you where that is. It's in Google Classroom. And so you'll just go to Classwork, Investigation 3. Read these directions, okay? But if I go to the lab map, okay, right here, if you click on this link, comparing DNA sequences to understand evolutionary relationships, that takes you to the very legal copy of the scan of the lab book. So you can read that, read that, read that. And the issue is at the point of publication, the directions for how to use BLAST, it's a website, were accurate, but they've changed since then. So I have updated, and so has the College Board. But look at the title of it, Comparing DNA Sequences to Understand the Evolutionary Relationships. So you're going to compare DNA sequence to say who's more closely related to who. This is a real scientist working lab site that you're going to be working out of. It is not a pretend, it's not mock-up for you in AP. But I encourage you, because there's really good information in the lab, to read through it. And you're going to be looking at a fossil from China. It has the coronavirus. Okay. And you're going to try to place it. She's drunk. Don't let her teach. You're going to try to place it here on this cladogram to see where it would fit best based on what you do in this BLAST lab. So that's that. The second thing is it's just a comedy of errors. The entire explanation that I would give you if I wasn't here today would be right here. If you go to YouTube, and then this video right here. Hi, this is Miss Litton. You know how it rolls. But this will explain all of the lab too, if you want to get a head start on that. The reason, what are you laughing at now?
You've seen yourself live.
What?
The live stream wasn't recommended.
Oh, okay. I didn't understand. Say it again.
Like the live stream wasn't, like the live stream that you're doing right now isn't the recommended.
Oh, how funny. weird. Oh, look, there's Emily again. All right. In a menopausal hot flash. Okay. So the instructions, you can follow these that I have printed in here, and then I tell you at this point, jump back into the lab book instructions because you'll need them there. But also the College Board has really good instructions there now. So I'm just going to show you. I'm going to go to the College Board website because what you're going to need to do is download 4 genes from the College Board website onto your computer. You will not be able to access that data. There's nothing on your Chromebook. There would be nothing on your home computer that could understand that data. It's a lot of data. And so I'll show you where those are. Okay. So you download these 4 genes, and you could do this at home. It's not underneath the umbrella of the OPUSD domain. So you could do this at home. And then after you download it, it says go to the BLAST site, and it'll tell you from there how to upload, once you've downloaded it, how to upload it into, and that's what BLAST stands for, how to upload it in there so you can analyze it. Okay, then your lab map, if you look, all of that is just instructions about it, okay? And then we get here, so you're gonna eliminate all of that because I don't wanna see my instructions from your team. And that's why I said eliminate all the prompts. Here's your driving question, I did it for you. On your background, there's 2 paragraphs. The first paragraph, I want you to address these 4 prompts, and this is what I want you to address in the second paragraph. Your hypothesis, remember, it's got to be if, then, because. I already did your variables for you and your constants. That's already done. Okay. Your flow chart, I'm not looking for detailed instructions. It's just that this is going to be separated because I'll have removed my instructions. You're going to do it very simple, you know, saying like you downloaded these. What's so funny?
I just keep thinking of Emily turning back, and each time she does, it just goes.
You know, people are going to be like, how many times was Emily in the video? Oh, and maybe we can make this look. It's amazing today, really amazing. There, we'll make the screen bigger. Okay. Come on in. I just keep failing, one thing after another. We're live streaming right now, so if there's anything you want to say, okay. You can't see? What? Okay. Okay, so your flow chart would be something like go to the College Board website, download the genes, upload them into the BLAST lab, and then what instructions you had after that. That's what you need to have there. Okay, your data table, I've already set it up for you, but there's information here you want to pay attention to in order for you to analyze the data. If you bypass that, you won't get the right thing you need. And this BLAST lab, this may actually not be true anymore, because they always get new information in it and it's updated, but at the time I typed the lab map, that was true. But you're gonna give the scientific name and the common name chicken and how close it is to, this was gene 1. At the time when I ran it, gene 1 was closest to similarity with the chicken. Okay, so you're going to do that for all those. Then you're going to answer these questions in paragraph form. You can leave the prompt there, leave the number 1. I'm okay with that. That way, because there's so many questions. And I even removed one question for you to make it faster, and then you'll do your conclusions. This is meant for you to do all during the class period and be done with it. But I don't know what your schedule is next week. If you're like, you know, I want to knock this out with my bio buddies are coming over and we're just going to get this done before Tuesday, be my guest. It's all there for you. You use your time responsibly for what you need to do. I have another POGL I'm putting out today for you to do, and that needs to get done. I'll probably say that POGL needs to be done by Tuesday morning at 8 a.m., just like the last one, to kind of help you stay accountable for that. All right? So that's what you're going to do on Tuesday. On Thursday, I'm going to see you. I'll be gone Monday, Tuesday. I'll be on Wednesday. I think Wednesday I have a review posted, maybe. Then Thursday, I have honors help with their lab, but also open if you want to see me, because I'm again gone on Friday and your test is on Monday. Okay? It's not my choice. So Thursday, we'll have reviewed everything as we went around, because I'll review on Wednesday. And then the last chapter on 19, or maybe we'll get really far, I've given you all the notes for chapter 19. They're completely filled in. The only thing you need to add in is the pictures. So as of today, you will have all of the content that you need for your test that's not next week, but it's the week after. Okay, so there is a student, I want to remind you of something. There was a student last night when we were doing advisement, and her name, I'm just going to say, it's, I don't have permission from her to say her whole name, is just Charlie, and then the last name is, do you know who that is? Okay, so she's really smart. Do you agree with that? Yeah, she's a bright girl. Okay, and I'm giving the talk to the parents last night at advisement about, the biggest problem is students need to listen to what I say, about how to approach the class, about studying ahead of time and not cramming, that cramming works for tests, but it doesn't work for the AP exam. And I said, the biggest problem I have is students just going, nah, she doesn't know what she's talking about, okay? And as a result of that, they get either low grades or they do okay in my class, but they don't pass the AP exam. And it was great because she was standing there I just pulled her up and she just started talking, and it was perfect because she said, that was me. Because she is a fantastic student, and she was doing what she needed in class. And she had her epiphany, as some of you have followed exactly what I've said to do since day one. You believed the words coming out of my mouth. Some of you just haven't, or you're too lazy to believe them. And Charlie believed it, it took her till December, and she goes, then I had an epiphany. And I was like, oh my gosh, there is a lot of material, because it had been accruing, right? And she's like, I'm not going to be ready for this AP exam. She knew she had not been doing exactly what I told her to do on her studying, doing a little bit every day, writing the essays as soon as you leave class, etc. So she had to start bearing down in December to work hard to review the material we had already learned. And I tell you this because her focus was the A. Her focus was, she just figured, I'm doing fine in the class, I'll pass the AP exam. She had never taken an AP exam before. She had never taken a science AP exam before. So instead of believing what I said, she just went with what had worked for her in the past. The reason why I bring this up is, as of today, you will have all the information for Unit 7. There's only one unit left, Unit 8, and then we're done. Okay, and that Unit 8, when we start that, it's on ecology, and that's great because you usually can relate really well to environmental things. You probably have a good working knowledge of it a little bit, so it's maybe easier to understand, and we'll talk a little bit about behavior. But at that point, Unit 8, you're going to want to have doing the reviewing as you're learning, right? Does that make sense? Do the reviewing as you're learning. Right now is the time to start planning for your review. It is not too late. You have all of March, you have all of April, and you have about 2 weeks in May. All right, so I want you to start going hard. I want you this weekend. Don't be random about, not a scattershot. We're dialing it in like a sniper, okay? We want to make sure that we cover all of our bases. What is the best documents you can use to review for your AP exam? Master Hyperdocs, baby, use those. But the first thing you should do is just not randomly like, I'm going to do Unit 2, okay? But this weekend, I want you to commit to a plan. I want you to commit to a plan because let's say one semester or another, you get a B in this class. And that's going on your transcripts, and that's what the colleges that you apply for, that's what they see. But there's something else they can see too. What else could they see if you say, okay? Your AP score. If you mic drop on the AP score and get a 5, and that is so hard to get. But if you mic drop and you get a 5 on the AP exam in biology, they're going to go, oh, they just had a rough semester. They got it together. Look, they got a 5 on the AP exam. Because that is what they can use to compare students across the entire United States. We do have a tremendous pass rate. You will beat so many, but they do. So many juniors and seniors from other schools, because most schools, it is juniors and seniors taking it, right? Whatever it was last year, 94% pass rate, and it's like 63, 64 nationally. So we're beating them by like 30 percentage points, and you're beating them as sophomores, and most of them are juniors and seniors with a full year of biology under their belt and a full year of chemistry under their belt and a full year of calculus under their belt. Okay, so I have confidence. We have evidence. When I say claim, evidence, reason, I can make that claim. You will be ready. The other thing is I just got an email. Remember how the tests have changed? And like I'm releasing, or the College Board, we can do those practice tests, right? And I release the frequently asked questions and the multiple choice. We've been doing that as we go along, yes? Well, I'm getting 3 full practice tests with the current version, and your teachers can get that too. They should have a note from the College Board, because some of them, they don't have access to any practice in their classrooms by their teachers. So I'm just telling you that I just got an email about that. So we have full practice exams that we can take, 3 of them, and one just got released, and then another one's going to get released in March, and another one's going to get released in April. So I think because now I'm going to get those, we might want to do one Saturday practice exam, one Sunday practice exam, and then one I'll just release for you to do at any time. The reason is you kind of get the feeling of it when you're all here together, and we can kind of go over the answers as well afterwards. So I usually have that on the schedule, but I didn't think I was going to have any current practice exams, but I just got some. Okay, so that's the plan there. Tell your bio buddy and tell your bio buddy at home. When will you need to come up with a plan. You've got 7 units, right? 7 units you need to review hard, and you start with the objectives listed at the top of the hyperdoc, and you just read through them and go, can I do that? Can I do that? Because the people who write the test, that's what they're looking at, okay? And then if you get to this one, you're like, I don't even know where that is. Go and look below in my chart where I tell you I've addressed that issue, and look at those group shared notes and review those group shared notes, or use the Bozeman's or the quizzes or anything else that's there to get you prepared for that. I will still, as we get closer to May, hold like a Unit 1 review, a Unit 2 review. I will still do that. But those will be a 7th period kind of fast and furious, right? You need to sit at your table, your dining room table, your desk, or sit on your bed and start reviewing that material, input and output, to get prepared. All right? So tell your bio buddy, when is that going to be? When are you going to make a plan? Yes? I haven't even written it yet. So I will tell you next week. Yeah. Now stop. If all your bio buddy did was give you excuses why they can't come up with a time, say that is unacceptable. You will do every day what is important to you. Get up a half an hour early and come up with a spreadsheet or a plan or something on your calendar, so you're going to review all the objectives that there are. If you wanted to, you could count up all the objectives and divide them by the number of days and hit that many objectives a day. That might be one way to go about it. But tell them, I want a firm time when I'm going to get your schedule. Or maybe you're like, this is my best friend, and so we're going to develop our schedule together and come up with a master schedule. Or maybe you want to do it as a lab team. Come up with a spreadsheet and say, we're going to review this by then. I don't care, but do something. Go, have that conversation. I didn't write it down.
We're at stage 4 evolution of cell.
Oh, okay. Oh, I didn't even talk about that.
We didn't get the notes for it.
Oh, but we talked about it.
Yeah.
Okay. Okay. Hi, Franklin Ding and twin brothers Asher and Jeremiah Kim from Anthony. Okay. We see you. I see you. Okay. All right. So we are on chapter 18. We already discussed the introduction and Luca. I'm combining Luca and introduction. And then we went over stage 1, how we would get monomers from inorganic molecules. Yes? And then stage 2, how those monomers would get together to form polymers. I'm going to let you review that on your own. And then protocells, remember, is when they have a barrier of some sort and they have metabolism going on inside. And then stage 4 is that self-replication system. And we went over that, but I didn't give you the notes for it. Is that accurate? Right on. Okay, so on stage 4, evolution of the self-replication system, RNA first, RNA coded for proteins, and then they hypothesized that reverse transcriptase, where have we learned that before? Good. Retroviruses, right? Form DNA as a storage location for the genetic code. Remember, that's that whole paradigm shift in the central dogma. Instead of DNA, RNA, proteins, maybe we started RNA, and from RNA we branched to proteins, and storage is DNA, right? Okay, and then protein first. RNA is complicated, so it's more likely that proteins evolve first to guide the synthesis of nucleic acids, And then, hang on one second, let me get here. Okay, or protein, RNA simultaneously. First cell would have true genes that could have been replicated because of the presence of proteins, since they developed at the same time. And remember, if they developed at the same time, that was because clay could act as a catalyst. Okay, and then the genetic code probably underwent natural selection to efficiently store genetic information. Okay, remember, we're looking at this whole chapter on origin and history of life through the lens of what? Natural selection, because that's what this unit is about, natural selection. All right, so ask your bio buddy, any questions, anything you don't get on that? Do you just need to review it? Go ahead, ask them. All right, anything you need to ask me before we start today? Do I need to do something? No. Oh, okay. All right. So we are at, if you look on history of life. So now that we have life, how did it change over time? And we know through natural selection, right? But the first part is fossils tell a story, and we've already talked about fossils, yes? Okay. And I pass fossils around. So the majority of fossils are found in what kind of rock? Sedimentary rock. And the assumption is the layers on top are newer, younger than the layers on the bottom. And you would have to watch out for a what kind of zone? Do you remember what that's called? Subduction zone, right? You have to lock them. And there's different boundaries, and we'll review those a little bit. You got all of that? Good. Now, I want to give you some perspective, okay, of change over time. So really focus up here, because this will probably help you. We're going to learn today the different time periods on Earth since its inception. When did Earth start? 4.6 billion years ago, okay? Now, at 4.6 billion years ago, we're going to say that was midnight last night. What were you doing at midnight last night? Were you sleeping or studying or gaming or reading? Okay. Hopefully you were sleeping. Okay? All right.
Watching Tasty 5-minute versus 5 minute.
Okay. Now, my suggestion is at 12 midnight, you should be what? Because your body needs it. Okay, now, midnight last night, formation of the Earth. So that would be when? How many years ago? 4.6. 1 a.m., 2 a.m., between 2 and 3, Miss Litton got up with her first hot flash, okay? Then 3, then 4. We're right now in the Precambrian Period, oldest rocks. Here, oldest fossils. Before that, 4:15, Miss Litton got up to exercise. Were you with me? No. Okay. Anybody get up or run or do something this morning? No. Okay. Ran to the bathroom. Okay. All right, it was really fun. Okay, now we have our oldest fossils, and we're still in the Precambrian period. At 6 a.m. this morning, were you up yet? Okay, first photosynthetic organisms at 6 a.m. Those first photosynthetic organisms, were they doing cyclic or non-cyclic photophosphorylation? Cyclic. So they were not generating any what? Yes, no reduced NADP, but also no what? Oxygen. That's That's critical, right? Yeah, it's critical. Because they weren't breaking, didn't have the ability to break or split what? Water. Okay, good. All right, so we do have autotrophs at this point. Still in the Precambrian period, 1 second equals 52,000 years. It's 7, it's 8, you're at school, 9, 10, 11, 12 noon. About this time, this is when the oxygen started getting in the atmosphere. So the Earth lived all of this time in what kind of environment would we call that? Reducing. If there is free oxygen in the atmosphere, plants must have evolved the ability to what? Do what kind? Non-cyclic. So they must have evolved the ability to break water, right? So that you could get oxygen and a source of electrons to excite, yeah? All right, so now we start to build up free oxygen. We're still in the Precambrian period, okay? Oldest eukaryotic fossils. What's the hypothesis that explains eukaryotes? Endosymbiotic hypothesis, mitochondria and chloroplasts going to live within the cells. Do we have evidence for that? Yes. Tell your bio buddy one piece of evidence. You can't say the same one. What were those free-living prokaryotes that came to live in another one? I heard you say their name. Mitochondria and mitochondria and chloroplasts have their own. It's in a, what's the DNA? A circle, okay? They're self-replicating through what process? Binary fission. They have their own ribosomes. They have a double membrane, so they could have picked up the second membrane when they got engulfed. Yes? Okay, so now we have eukaryotic cells, and what time is it? 4:30 in the afternoon. Okay, eukaryotic cells. So we're still only one cell big. Do you agree with that?
Yeah.
We went from midnight all this time. We're at 6, 7, 8. Between 8 and 9 is the first time we have anything that is what? Multicellular. Notice we are still in the Precambrian period. Okay? Now, about between 9 and 10, we're in the Paleozoic era, old and pale. And it says land plants. There's a lot of other things that happened during this time. Then Mesozoic era, age of dinosaurs, we're like somewhere between 10 and 11. Cenozoic, the era we're in right now. And not until tonight, 30 seconds before midnight, would humans have arrived on the scene. Okay? Just for perspective. Okay? So on your notes, go to your notes. 24-hour Earth diagram illustrates that only single-celled organisms were present during 80%, okay, 80% of the Earth's time. 12 midnight is when what? What would you put there? Earth forms. 5 a.m. you have your first cell. Good, first cell. And it's a prokaryote. 4 p.m. we have eukaryotes. 8 p.m. we have multicellularity. 10 p.m. we have invasion of land. We're getting out of the water, we're getting land. And then at 11:59 and 30 seconds, who shows up? Humans show up. Good. Okay, now I showed you a bunch of fossils. I'm just going to run through a few for you to look at. Okay. Do you remember this guy right here? Trilobite. Good. Now, right here at the junction between Precambrian and Paleozoic era, at the very first period of the Paleozoic era, which you ready for its name? Ready? Cambrian. So does that make sense why everything here is called what? Precambrian. Okay? That's when you got trilobites. That's when trilobites showed up. That was a fossil that somebody had broke, right? Some past, not anybody in this class. Yes?
Weren't the first things that formed?
Yes. But that's just an index fossil. It's easily fossilized because of its exoskeleton. So you know if you have a layer of rock that has a trilobite in it, any layers below it are older and probably Precambrian time. Any layers above are in the Paleozoic era sometimes. Does that make sense? Okay, or more. Good. Okay, so we did that. Youngest bio buddy, we're just going to ping pong back and forth. Tell them what you see and what you know about it. Would you find a woolly mammoth in amber? No, he's not going to be like stuck to a tree. I can't move, you know, and then sap gets on him, right? So you're not ever going to find a woolly mammoth in amber. Only very, very what? Small things, okay? And go again. Could you find him in ice? Yes. Okay. Look, that looks like teeth to me. Okay? Then do you remember how I showed you those clam fossils from my family's ranch, how they look like mold? Remember I said about a cast, right? Do you remember that?
Yeah.
Okay. All right. And then here is a imprint. That's a big, big fish. All right. Now, how do we know how old those fossils are? That is one way. And there's another one too. I agree. Not it. Differentiate, please. Relative, like when you can't find anybody else to go to the prom with you, so you ask your cousin.
Oh.
All right, keep going, keep going. Cousin Levin. Oh my gosh, we're recording. All right. What do you see right here? Trilobite. And what is he? Good. All right. And you use a combination of relative and absolute dating in order to maybe identify rock layers. Whoever explained this, I would like the other bio buddy to go in more detail. Please go. All right, now, most carbon that you see is carbon 12. That's why on the periodic table, I believe it's like 12.011, I think is how it is on the periodic table. And that's because it's an average of all the isotopes. There's carbon 14. Carbon 14 doesn't break down into carbon 12, it breaks down into nitrogen. But currently, if you picked up a rock today that was formed like in Hawaii yesterday afternoon, you would see a ratio of one isotope to another for carbon. So I'm going to use this as an analogy. This is not to scale, okay? This is not to scale. You can't see. There's a white paper there. See the white paper? All right. So the white paper, what do you think that represents? Guesses? Carbon, which one? 12 or 14? 14, right? Because we see less carbon 14 than we do what? Carbon 12. This is if I picked up a rock today, okay? If I'm digging, pay no attention, I'm digging for a fossil. Oh, I found one. Here's my fossil that I found. Now, is it the same ratio of carbon 14 to carbon 12? No. So I take and I look, that's one half-life, 5,700 years, right? Here's two half-lives, another 5,700 years, and another 5,700 years because I know the rate of decay of carbon 14. Does that make sense? Okay, so let's go to our notes. So besides sediment, fossils can be found in tar, ice bogs, you got all that. Dating. Relative, older layers at the bottom. Index fossils, Slate, finish up where it says about index fossils. You explain it. Identify deposits made at the same time worldwide, and that's what they are used for. Oh, that was not my face, sorry. Okay, absolute dating relies on radiometric techniques. It's the speed of isotope breakdown is what you're based on. And radiocarbon dating, I think that Blue can explain that. Can you explain that to the camera? You can just talk, they'll hear you.
Radiocarbon dating, 14C decay measured 5,700 years ago to become 14, and compared amount of 14C to 12C currently determined age. So radiocarbon dating is when you... Oh, there's 3 people. When you're...
Okay.
Oh, I'm not on it.
No, I know. Okay, so I thought you might find this interesting. This is just some different isotopes and how fast they decay. So you wouldn't want to use bismuth-210 because its half-life is how many? 5 days. That's like how you would measure how old a peanut butter and jelly sandwich is, not how old a fossil is. And then you can see some other ones are, not even a second. Okay, so you have to pick the right isotope to look for how old your rock is. Okay, that is it. Now, I think, yes, great. So next, okay, take a look at this diagram. Got it? Actually, that doesn't matter. I want Slate to do all the odd numbers. Tell him anything you know about that, because I need you to start pulling from units 1 through 7, not just 7, okay? Slate will do the odds, Blues will do the evens, okay? Do that. Who wants to? Here, we're going to listen to you guys talk about it. Oh, here, I'll still put it on the screen. Here. Or talk loud, or come over there so they can still see the picture. Go over by that so they can hear you talk.
The first cell happened,
Because they need to see the picture.
and then it branched off to archaea. And that was like a split. And then archaea
It's a mess. You love it?
made, they made the, you have to go.
I did bacteria.
Eukarya.
Eukarya. They're organisms with membrane-bound organelles.
And then a type of eukarya is heterotrophic protist. And then mitochondria went inside the heterotroph. It like engulfed it. Yeah. Endosymbiotic hypothesis.
The same thing for the photosynthetic process.
And then the mitochondria moved on.
Okay, come back to me. Yes.
Isn't that wrong because you said that like archaea came first? Or it came later?
Oh, you mean because it looks like they split at the same time? Yeah, I don't like that either. Yeah, so you have bacteria and then archaea. So I wish it would like do a little something there, but it didn't. I agree with that. Okay, so this gives you an outline, okay, a good outline. I really like this diagram. What I'm going to teach you right now is I'm going to teach you the time periods in a way that hopefully you will never forget them the rest of your life. And then once we have our scaffolding set up, then we'll just fill in the details a little bit to make sure we understand it. The goal is going to be not to memorize every single organism or every single time period. Our goal is to know the names of the time periods. There's only 4 that I need you to know, okay? And where one stops, another one starts, okay? And then the bookend organisms, because if you know the bookend organisms, you know everything that comes in between. Like if you look at our schedule today, right? You knew that let's see, the rally got out at 12:20 and this class started at 1:05. What did you know have to come, or 1:07? What would have to come between that? Lunch, right? So if I can teach you the bookends, then you'll know everything that comes in between, okay? Now, I don't know how else they can see me because I want to move across this board because that's how I have to do it. So maybe you can, like, look at it.
Like moved?
Yeah, maybe go back. So go back so I can see this whole front. Otherwise, I won't know how to explain this because this is how I explain it.
Is this what you mean?
Okay, let's see.
You can't see anything.
Whoa.
Okay. Yeah. Oh, no. Okay, so, wait, come closer. You're too far away. Come closer. Come closer. What's the comment? What is extremely helpful, Mr. Jeremiah Kim? Who's Jeremiah Kim? Jeremy, but Jeremiah. Okay, are we ready? You can tell her if she doesn't have me in the thing. Okay, so the era we're in right now. The era that we're in is called the Cenozoic Era. Say it. Cenozoic Era. It's the center of your world right now. It's the center of your world. Now, it's this second. Now it's this second. Now it's this second. And it goes back in time, all the way back to 65 million years ago. Now, what's the center of your world right now? Maybe learning how to drive. What is the speed limit on the freeway? 65. So you're going to remember 65. Does that make sense? Wow. With me? You with me? Stay right there and just kind of turn a little bit. All right. So tell me, what era are we in right now? Cenozoic. And it starts right now, and it goes back to how long? 65 million years ago. We're going to remember because we drive. Okay. When the Cenozoic era started, there was another era that just finished, and that's the Mesozoic era in the... What did we normally do? We did here. Chromosomes meet in the... Thank you. Mesozoic era in the middle. That was a hard pull. Okay, Meso in the middle. Now the Mesozoic era ended 65 million years ago, right over here, 65 million years ago. Okay, but it went back in time till about 242 million years ago. Notice I'm in millions, right? 242 million years ago. What happens around 242 every day at school? That's when you, yeah, school's over. Okay, school's over. So that's the Mesozoic era. Now the Mesozoic era is broken into 3 periods. I'm not having you learn all 3 periods. I'm just going to tell them to you right now because one of them you already know. There's the Triassic period, then the Jurassic period, and then the Cretaceous. Now the Jurassic period, what are we going to think? Dinosaurs. Yeah, dinosaurs. What you're going to see as we talk through it, as one organism gains ascendancy, then another one starts evolving, and then it gains ascendancy, and another one declines, and so forth. So when you're in the middle of the Jurassic period, it's all about dinosaurs, okay? So what was the first one? Cenozoic, and it goes back what? Let me just tell you right now, the Cenozoic era is all about mammals and flowers. Okay? Mammals and flowers too. If you're driving your car for a date, then you're going to give your date some flowers. Yeah, and you're going to date a mammal. Okay. Hopefully a Homo sapien sapien. Now, so the Cenozoic era ended how long ago? 65 million years. And then what era are we in? Mesozoic. What's big here? And Mesozoic era started when? 242, because we're going to remember what? School. The era right before that is Old and Paleozoic. Say Paleozoic, okay? Old and Paleozoic. Now, the Paleozoic era goes back in time to about 542 million years ago. What are we doing about 542 every day? We are what? Eating dinner. So all you have to remember is drive, school's out, and dinner. And then you have everything. Because this right here is the Paleozoic era, and the very first period in the Paleozoic era is the Cambrian Period. What's the time right below that? Precambrian. Precambrian ends at what point? I just gave it to you. Think dinner time. Any time close to dinner. 5:42 dinner time. Now, when did the Precambrian era start? 4.6 billion years ago. So these were all millions, right? I'm at 542 million. Now I've got to get all the way back to the billions. I would have to run up to Medea Creek, okay, and maybe a little bit farther to represent for you the start of time. Because 87% of the geologic time scale occurs before the end of this carpet. 87% is Precambrian time. And these names, Precambrian, Paleozoic, Mesozoic, Cenozoic, it's not like they set it out and said, this is some time and it was written. Those are all man's ideas. And the reason is, the very first period of the Paleozoic era, there was a big change. It was the Cambrian explosion of life, of plant and animal life. Okay? Then by the time you get to the Mesozoic era, you're starting to get all these reptiles and even the beginnings of mammals. And by the time you get to the Cenozoic era, you're full-on mammals and flowers. Perfect. Okay? Not it. Tell them what you know. Tell them what you know. Hope that was helpful.
Is divided into 3 different ones. Jurassic, Triassic, yeah. Triassic first, Cretaceous, and then you go back to 542. Yeah, 542, and then.
All right, come back to me. Look at the Precambrian period on your notes. What does it say for the time period? 4.6 what? Billion years ago, which was the start of the Earth. Was there life on Earth right at 4.6 billion years ago?
No.
No. Okay. It took another what? 1 billion years, right? Our first fossil is about how old? Yeah, 3.5, 3.6 billion years, somewhere in there. Are you following me? Okay. And then it takes us all the way up to what time? 542 million. So keep your millions and your billions straight, okay? So number one is 87% of the total geologic time. Your first cell was about 3.5 billion years ago. Okay, now I want to show you some pictures. Okay, take a look here. This is showing you what's going on, the big ticket events in the Precambrian period. Why don't you ping pong back and forth? Slate, you go first. Slate blue, slate blue, slate blue. Go. Oh, yeah.
Oldest known fossil, 55 million years ago. Go, you're fine. Oldest eukaryotic fossil. No, these are the trilobites.
Are you ready for me? Okay, so one of the things you see is, you know, the oxygen revolution is huge because this is changing our environment from a reducing environment to a... Okay, good. So cyanobacteria, okay, those guys are pretty cool, right? What else do we know about cyanobacteria? Photosynthesis, yes. What kind? Cyclic, non-cyclic? Non-cyclic. They generated oxygen, okay? Do you remember about nitrogen fixation? Yeah. We talked about that a little bit. We're going to learn about that in Unit 8 too, okay? But cyanobacteria are bacteria, and obviously, and they're doing photosynthesis, but where they live, they would start out, you know, just a small group of them, and they create kind of a slime. And they're doing their photosynthesis, they're generating their oxygen, but the ocean is right there beating against them, and calcium carbonate, like these salts, are embedding in their bacteria and in their slime, and as a result of that, it kind of hardens, so some bacteria get left behind, and they are what? And some are on the edge. They keep moving outside of their calcified slime, and they keep moving out and keep moving out and keep moving out, and these right here still exist today with bacteria on them doing photosynthesis. And you know, like if you cut a tree down and you could see the rings and talk about what has happened over time during those different rings and what they represent, same thing here, but not using tree rings, you're using bacteria. And it's telling you a story as you go back through. So those are called stromatolites, and I don't know whose turn it is, but you do. And talk about those stromatolites. Go ahead.
Cyanobacteria that have mucus and gets caught in the mucus, calcium carbonate in water and rocks. And keep cyanobacteria can be eaten outwards and the cyanobacteria contributed to the oxygen revolution.
Okay, now I want to show you a video about the criticalness of oxygen, the oxygen revolution. So yeah, half-mast your Chromebooks, please.
Of all the mutations that lead to human life, the most important is also one of the earliest, the jump from simple single-celled life to complex multicellular life.
There's a lot of good information in here to use as
Hard
review.
to imagine
So
without
listen.
multicellularity, intelligence is possible. You've got to have a big enough organism so that specialization is enough that some of the cells, some of the fraction of that organism can just focus on being a brain.
The jump from simple life to complex life begins around 2.4 billion years ago. What was the trigger for this revolutionary change? Scientists have boiled it down to a single game-changing mutation: photosynthesis.
What happened was a mistake. A mutation led to a true superpower.
The first bug to develop this superpower is called Cyanobacteria. It turns green and is now able to use sunlight to create its own food supply.
For the first time, life had its own internal energy source. Wherever you are, as long as you have sunlight, you have food. So you could cut the umbilical cord and break free and start to really branch out into the environments of the earth.
The cyanobacteria thrive.
Wait,
But the waste
I have to pause because they're saying we've detected copyrighted audio, but I'm not making money off of it, so I thought it would be okay. So I need to pause you. I'm going to pause you for a minute, and then we'll continue the video.
The YouTube algorithm is scary.
Yeah.
Quick, add a reverb to it. Add a reverb.
Yes. So we'll be back, we promise.